Okay. All right, guys. Let's look at slides and see. Um, of course, this is this is not actual sections of anything, but is you remember this slide and then somebody shows you pictures of one type or the other of the different types of muscle, I guarantee you that you're going to have the probability of distinguishing which one is which, all right? Which one is the one that everything is looking straight, which one is the one with the funny looking arrangements in there, and which one is the one that doesn't have anything, it's not striated, okay? So that I included this again as a scheme to remember. And when you look at them from a section point of view, in the skeletal muscle, you're going to see always the nuclei in the periphery, all right? And when you do a heart, it's going to look pretty much the same. It's going to be muscle. It's going to be a muscle-looking structure, but there will be the nuclei are going to be, whenever it's visible in that particular section, it's going to be centrally located, all right? And if it's a longitudinal section, you're going to be able to distinguish between the homogeneity of that picture and the kind of funny-looking heterogeneity in terms of the cellular limits, boundaries between one and the other. All right? Skeletal muscle has to do with the skeleton. All right? So you might actually see pictures in which portion of the tendons are actually participating in this. And somebody shows you a picture of the famous Golgi tendon organ, you're going to be able to distinguish the existence of some portion of the tendon along with portion of the muscle. All right? So remember, this is skeletal muscle. So it's very easy to tell right now which one this is. Homogeneous longitudinal fibers with typical striated segments and the nuclei in the periphery. So this is skeletal muscle. We look at the skeletal muscle in a transversal section at different levels of magnification, and we see, in any case, we see the nuclei, they're always in the periphery, okay? Nuclei in the periphery. Nuclei in the periphery in every case. And what we find now is the connective tissue that appears in white. And we know that the different levels of connective tissue, the famous epi, peri, and endo layers of the connective tissue, all right? So each of these things would be microfibril, and therefore these microfibrils are forming a particular bunch together. Questions, by the way? I forgot to ask questions. For questions in the previous two ones. All right, everything is clear. This is a very nice picture, very organized picture of the structure of the sarcomere from Z to Z with the A band that is primarily composed of it's given this darker, stronger density because of the presence of myosin. Then the H band, which is this lighter portion inside, which has the M line right in the middle. You can see there is you can distinguish these little things that constitute the M line, and that is where the myosin filaments are actually bound to, they're anchored in there. And then you distinguish the I band, and in the edges of the I band, in the middle of the I band, over the C line, you can see structures like this, where the centrally located portion is the T-tubule, and then there are the two portions coming, which the T-tubule, as you I'm sure remember already, comes from the sarcolemma. It's an invagination of the sarcolemma, and these two other vesicle-looking structures, which are called the cisternae, coming from the sarcoplasmic reticulum. So this is the entire structure of the sarcomere. Questions? Another view of the same thing. This time the emphasis is placed more on the actual tubules. This is a triad. We need to recognize what a triad is compared to the previous slide, and actually even compared with the ones on this side in the I band. This one is very, very easily to distinguish between the T-tubule and the two cisternae coming from the sarcoplasmic reticulum. As you see, there's plenty of mitochondria in there. And then we can, again, separate and distinguish the A band and the I band. And these other vesicles, which are by themselves kind of in the middle of everything, correspond to the sarcoplasmic reticulum that has been sectioned at different levels, depending on where the section was located. Questions? You're going to get tired of looking at triads and sarcomeres. Going to have nightmares tonight. Sarcomere was coming to the room, something like that. So again, this is actually even try to use a scheme just in case you haven't learned so far what these things are, where the location of the sarcomere is and the triads and the arrangement of the tubules, the two tubules, and which are coming from the sarcolemma, I guess, and the other portions which are coming from the sarcoplasmic reticulum is 1 and 2. And as I said, in the cardiac tissue, there is 1 and 1. Okay. This is again another picture of the same kind of arrangement where the different bands are identified, but this time instead of putting them with name, which every number of everything is here, but we all can identify what it is. The only thing that we haven't mentioned is where some of the accumulation of glycogen can be located, and it's typically located in the surroundings, in the surroundings or the sarcoplasmic reticulum, so it's typically located along these potential locations. Everything else, if you look at by yourselves later, is exactly the same description of things. The C lines, the I band, the A band, the H band, the M line, and so on and so forth. And I guess you can figure out what number 6 is. Have you ever seen a triad before? All right. Questions? In case you still haven't seen everything about it, you have now to see the same structure in the different locations. These are a bunch of mitochondria in there, and everything is about the same. In this case, number 8 is located in this area, which is where, as I said, is glycogen, which accumulates in or in the proximity of the sarcoplasmic reticulum. Everything else has to do with the structures that we have already been relating to. Okay, and just in case you were wondering, there is a triad right there. If anyone imagine there is a question about triads in the exam, and I find that any of you guys have failed that question, I'm coming and personally make you do 50 push-ups. Okay, so you exercise your muscle. All right. As long as I don't have to do them at the same time, that's fine. All right. So this is now transversal sections at different levels in the structure of the sarcomere. As you can see here, we're looking at a portion in the sarcomere where there is the thick and the thin filaments are located. And we can see the thick filaments, and if you guys have the patience to count, you can see that for every single dark dot in there, there should be 6 of the thin ones around it. That's the perfect view of the structure. Now, sometimes the perfect view gets slightly distorted, and I want you guys to know about that, because when you do the sectioning, the cutting of the tissue from the organ where you're coming from, and the sectioning by the microtome, and all these kinds of things, occasionally there is some pulling rather than a perfect cut section. So in some cases, the structure is not as beautiful as in the books, but you can see that there is, easily you can see that these are portions in which you can see the 2 types of filaments very easily located. Okay. So again, these are typical structures. There is nothing new that you haven't seen before, the mitochondria, the sarcoplasmic reticulum, and simply this is a way of showing that we are in the middle of the sarcomere somewhere where there is the overlap between the 2 kinds of filaments. Questions? All right, so this is a transversal section. And again, and we are seeing here, this is a beautiful example. I'm sure whoever did this slide had to spend some time looking for the perfect one, and here is the perfect one. A myosin filament, and 1, 2, 3, 4, 5, 6, perfectly distributed, 6 thin filaments of actin. Right? Finding the same perfect structure as I just said in other cases is not as clear as it's not as perfect in many cases because of the way this sectioning is done. Questions? All right, so again, to refresh our memories, if we cut through a portion where there is both filaments, we're going to find these kind of structures in either side. And if we cut through this one, we're going to see just the myosin by itself. If we were to cut, and that is not shown in this scheme, if we were to cut through the eye band around there, we will see a picture like this, but all with thin filaments, as opposed to the thick ones. Okay? Questions? This is another way of showing you guys the same, and this shows more like the imperfections that I was talking about. Things might be a little bit also disorganized because when you cut a section, I suppose you guys have studied all the histological techniques and all that stuff, and they're masters in the system, right? But to be able to do the sectioning and the staining and all this, one of the first things you need to do is actually put the sample of the tissue most of the time in something as nasty as formaldehyde. Okay, there are newer and nicer things that people use these days, and in fact, it's great because some of us who wanted to be biologists many years ago and used to put even animals, full animals inside formaldehyde, and had formaldehyde in my house, and I was exposed all the time, and developed a hypersensitivity. So now, if there is anyone with formaldehyde in the building across the street, I start crying, okay? And I don't like to cry, but especially in public. But what I mean to say is that formaldehyde is not just holy water, okay? It does lots of cross-linking things and maintains proteins bound to each other in a very nasty way. Then, in case that's not enough, then you're going to now have get the thing and put it in something like paraffin or something to give it consistency. So paraffin is going to be getting into the tissues. And then, if that is not enough, in order to do the staining, you're going to now remove everything, deparaffinize, heat the samples. In other words, get rid, dehydrate the samples with cereal, alcohols, and all those kinds of things. It's a whole process that really doesn't maintain faithfully the structure. This is supposed to be, however, for the EM. The EM doesn't use any of this stuff. It uses a number of other things which are particular, again, they're not holy water. So they might modify the structure. So if you had the time, and I'm sure you don't have to study anything else, you have plenty of time, just spend some looking at the little filaments and see how many you see, because you can see in many cases more than 6. And I spent some time trying to do that, and in the majority of the cases, you see more than 6 because of what I just said. It's the disorganization in some parts of the slide. But this is what you will see in what kind of region? In the region where there is overlap between the two filaments, the myosin and the actin. Okay, questions? All right, so this is, we've seen a picture similar to this one in the class, in the lecture. This is the way the nerve is coming to a motor unit, and how this formation of the so-called motor end plates, which are where the synapses are, and how the signaling is transmitted. So these kind of buttons plugging into the muscle fiber, and they're going to be able to direct the movement of the fiber itself. Questions? This is now a motor end plate being looked at from the EM perspective. This is the axon that's coming. This is the structure of the presynaptic portion of the synapse. This is the synaptic cleft, and as you remember, in the case of the muscle, there are these particular folds, very kind of interesting arrangements of the membrane, and some of the invaginations of that is going to form the T-tubules that will be the first one catching the wave of the signal and transferring that signal to the sarcoplasmic reticulum, where the calcium is going to be liberated. Okay, so these T-tubes that are formed at this level is where the action potential is transmitted into, and how the signal is transferred. Okay. So this NT means nerve terminals, and these are all mitochondria and different other organelles that are common to most of the type of synapses. Oops, sorry. Questions? All right. Spindle, muscle spindle, right in the middle of the picture. It's smaller than the arrow, just in case anybody misses it. Okay, but what we can see is the inside, the intrafusal fibers, and by definition, anything that is not intra has to be extra. So the extrafusal are all the other ones, are doing primarily the motor job, and the intrafusal are primarily doing the sensory job. And it's a equilibrium between the sensory receptor, the message received through the sensory system that is going to be somehow translated at the level of the central nervous system and converted into whatever action is particularly requiring response. Questions? In case I am very bad about explaining what these things are, there is a scheme trying to tell you exactly how things are organized. There is a kind of membrane capsule. There is a place where there is communication with the outside and where the fibers, nerve fibers are coming in and out, and the intrafusal fibers and the extrafusal fibers. Okay, so this is primary a sensory organ, a sub-organ, if you want, within the muscle tissue. This is another vision of this, another view of this. You can see the intrafusal and the extrafusal. But I want you to realize one thing. Even the intrafusal, and even though, remember we said that there were two types there of the intrafusal ones, the ones that had the nucleus in a kind of a chain, and the ones that had the nucleus kind of in a bag. The position of the nuclei is, in general, guess what? In the periphery. Okay? So even if someone were to be nasty to you and show you just the intrafusal in an exam, just the intrafusal transversal section, and say, what kind of muscle is this one? You could immediately say that this, because of the location of the nuclei, is a skeletal muscle. All right? I'm not that nasty, okay? So don't expect something like that from me for the exam. Questions? I'm sorry. In case you haven't seen a heart up close, that's what it is. It typically is the size of a fist, and is hard, works very hard through our lives. And the top two chambers are the atria, and the top two chambers inside are the ventricles. And the conductivity goes from the top to the bottom, and then is spread through the ventricles itself to allow the contraction in this fashion. You can see now the structure of the heart muscle, which is, it gives you the impression of being fairly disorganized after you have been looking at the skeletal muscle. This is kind of messy. You can see that there is all kinds of structures, kind of spaces in between. There is a separation and branching in different locations. There is a very different organization. So if someone is to show you two pictures of one and the other next to each other, it's going to be very easy to identify which one is which. Questions? By the way, if you look at this one and you don't find any nuclei, don't think that anything happened to this heart, poor guy, okay? It's simply because it doesn't stain the nuclei. It just stains the cytoplasm or the sarcoplasm. In this case, everything is shown, and again, the main properties or characteristics of this is these funny-looking structures that are kind of step-like structures in different positions that mark the separation between the different fibers. The shape of this fiber, for instance, is kind of funny compared to this other one that looks more like a straight type of fiber from the skeletal muscle. In any case, the nucleus is centrally located, and an example of branching, another example of branching, and this one actually happens with the intercalated discs. When you look at references in the literature about the discs, 50% of the population use DISK, and the other 50% DISC. So call it whatever you want, write it whatever you want, it means the same thing. Questions? All right, so the purpose of this is to show you, again, the irregularities of these. These are different cells cell 3, cell 4, cell 5, cell 8. So the situation here is that we want to show the separation between these two cells. This is an intercalated disc. You can see following the arrow. There is a vertical portion, there is a horizontal portion, and there is another vertical portion. So it's always a kind of zigzag type of organization. And as we looked at during the lecture, the structures that you find in the vertical portion are not the same as you find in the horizontal portions. In the horizontal portions, primarily what you find is gap junctions. In the vertical portion, you find this fascia and the desmosome, which are the ones, the tightest kind of unions between them. Questions? Yes. Capillary. I'm sorry? F is actually an ending from a nervous fiber. Nervous fiber ending that has been cut before this level of the synapse. Oh, I'm sorry, I'm going backwards. I don't want to do that. All right, so this is the cardiac muscle. See the prominent mitochondria, larger, thicker than that. The sarcoplasmic reticulum vesicles. But if anybody finds a triad in here, I'll give you a Nobel Prize. There's no triads in the muscle, in the cardiac muscle. Remember, all you see is dyads. It's one microtubule with T-tubule with one vesicle from the sarcoplasmic origin. Questions? Yeah. It's probably easier to see. They have to be in the same location as the other ones. Remember, they were always on the Z line. So they could be these two vesicles in there, maybe a dyad. I should have actually marked some of them before and looked at them more carefully. But there are simple two vesicles in the same location that the triads are in the skeletal muscle. Right, so again, the purpose of this slide is to show you the intercalated disc. In this case, I'm going to be following the line where it is located. It's kind of going all the way around, turns here, makes a left turn on the other side, and goes all the way down. So in this vertical portion, this would be the prime example of the so-called fascia adherens, is FA. All right, in the horizontal positions, wherever there is an N, is a gap junction, and these other portions in here, which are actually also present at high density in this other location, where it says D is actually desmosomes, and these are mitochondria, and the SR is sarcoplasmic reticulum. Okay. I'm sorry, questions? Where is G? Oh, this is, remember that the regions where glycogen is accumulating? Okay, sorry about that. Oh, going backwards again. Okay, so this is, again, another structure, and the idea is to see the junction between cells once more. You see this tortuous road that is going around here. It's going all this way, again, simply to show you how heterogeneous, in a way, irregular the junctions are between the cells. The staining allows for every word where there is a desmosome to have these dense structures, and those tend to happen in the vertical portions of the intercalated disc. And this is an example of what you see in this section, the myocardium of, in this case, in particularly of a guinea pig. Questions? This is to remind you guys of the conduction system in the heart. The sinoatrial node, which is the one that is like a pacemaker, could beat by itself if you are brain dead at a rate of 50 beats per minute. Transfer the main impulse to the atrioventricular node, then the impulse comes down through the septum between the two ventricles and is spread thanks to the so-called Purkinje fibers spreads to the rest of the wall. So the first wave of contraction happens at the top and the bottom part later, and the separation between the different cavities, as you guys know, doesn't allow for the mixing of the oxygenated blood and the deoxygenated blood. Questions? The idea is mainly to show you the location of this specialized conduction system. Picture of Purkinje fibers. Okay. Again, if we were to identify this slide is in the reverse position relative to the one that I showed you during the lecture. So this is the endocardium, and these are the Purkinje cells, and these are the actual endocardium, a myocardium that contains all the actual real typical muscle cells from the cardiac tissue. Questions? This is the representation, actually it's an actual picture of an atrial portion of the heart, showing all these granules, dense granules, when you stain them for the electron microscope, that contain the AMP, the atrial natriuretic peptide, that is secreted and, as we discussed during the lecture, participates in controlling blood pressure by affecting the kidneys once it gets there, to promote the release of water and sodium. But you can see, you can use this picture also to look at the overall structure of the muscle fibers, and then you can see everything we've been talking about before, the irregularity of the divisions and the step-by-step separation between cells. Questions? This is now the smooth muscle. This is indicating, presumably indicating, that is the wall in the stomach. We'll see that when we look at the GI system on October 18th. And the typical structure of the relaxed smooth muscle cell and the contracted muscle cell with the famous twitched arrangement of the nucleus, because it's the only way to solve the problem of not become enucleated in the process of contracting. Typical longitudinal section indicating this spindle-shaped cells with center nuclei. Typical transversal section indicating, of course, you've been lucky enough, some of the cells will look the nucleus. In any case, it will be centrally located, and it will take more or less space between the circle of what is the cell, depending on where in the cell you actually perform this section. Right? Questions? Now this is what it looks in the case of the urinary bladder. The tissue is the smooth muscle cells. This is a primarily longitudinal section. You can see here almost an entire spindle-looking cell, another one right here, the elongated nucleus in the center of the cells. Cells are nice and tightly bound together, and there's plenty of gap junctions that are communicating each other along with the reticular cells, and the electric stimulus for the contraction is transferred from one cell to the other through the gap junctions. Okay, so in that regard, they're closer to what an electrical synapse was. Remember, the electrical synapse was more like tight junctions between the pre and postsynaptic membranes, and that's the way it was transmitted. So this is more typical of the way the smooth muscle cell contracts. Questions? Is same tissue, lower quality picture, same idea, longitudinal section of the muscle cells with the nuclei in the center. Questions? This is now a section of the entire small intestine. You can see all these structures which are formed by epithelial cells. This is pretty much where all the absorption and the metabolism of the nutrients that come this way are located. There is a layer now of the smooth muscle cells, and then these are going to be making kind of peristaltic movements as the foot comes along to force it going in the proper direction. And then there is a membrane that covers parts of it. We'll look at that when the GI. But this here, I want to make sure that no one gets, if you were to get this picture, which I have no idea whether you'll get it or not, it's not up to me. This is not a nerve ending. Is being in that position to stimulate anything for this cell. This is a portion of that membrane, the mesentery membrane that is part of the linings of the way the gut is organized inside the limited space that we have in the cavity. That is called the mesentery, and that's the mesentery portion that has been remained with the preparation. This is not a nerve ending, even though it looks like it's kind of one of those button things, and it might even have physical-looking stuff inside. Okay, it's not. Questions? All right. So that's now the different portions with the proper names that we will look at later when we look at the GI by itself. Smooth muscle, and this is one portion which is longitudinal. This one here. And these are transversal sections. So the way the preparation has ended up is showing you in the same slide, both of them. And at the very bottom, there is a mixing with the connective tissue. Questions? This is in a blood vessel. You can see the erythrocytes right there. You can see the connective tissue out there. You can see all the smooth muscle with the elongated structures, the elongated cells. And I've been thinking about it, almost losing sleep. I don't understand why the nuclei appear the way they look. Maybe the person was kind of nervous when he did the preparation. But that's the way it looks. It's a densely packed portion of smooth muscle. Okay, that forms a substantial portion of the wall, in addition to the connective tissue inside and the endothelial cells in the inner part of the vessel. Questions? Another view of smooth cells in the vasculature. Okay, this is the only erythrocyte that was caught in the act of being there. This is the many folds that you can find that the endothelial cells are making, and then the cells around the vessel in the same organization as the kind of was seen in the previous case. Questions? Same thing. Nuclei is important to see that there are cells that are also some of those reticular cells I've been talking about. Some of the mitochondria, the nuclei are located in the right portion, and some of the gap junctions that exist between the cells that allows the communication and transmission of signals from one source to the neighbors. Questions? The arrows are pointing. I could have used this as an example of gap junction. There's another one on the other side. Again, a smooth muscle cells with the different number four here and there. Those are nerve terminals. Those are the connections with the nerve tissue. Of course, number one is the nucleus, and then there is a different kind of connections. Number three is tiny little things that you can see inside the cells. You can see them in some that are not being shown in the number 3 with the arrows. Portions close to the membrane, which are glycogen particles. The glycogen accumulated inside the cells is also a potential source of energy. And the number 2 is supposed to exemplify the sarcoplasm. So this is just the cytoplasm that is only visible This is a portion of the cell in which there is no nucleus visible. So everything else, except for these little, the kind of darker dots, which are the mitochondria, is just the other components of the sarcoplasm. Questions? And you have seen this one before, and you need to know what the caveoli are, especially because Dr. Notario used to work when he was young on caveoli. Okay? Remember, he was young at some point. By the way, this is the junction, one of the junctions that we've seen before in other micrographs before that were pointed by arrows, but this is in a much more detail, the gap junction between the two cells where the transmission is done, and SR is the sarcoplasmic reticulum, and M is the mitochondria. Questions? Thank you so much. I'll see you guys in 2 weeks precisely. I will enjoy visiting for the first time my new grandson. And yeah, he was born just earlier, but we haven't met yet. I'm going to teach him some histology first. Thank you.